Thank you, everyone, for having me. Yeah, as you might gather from my title, my talk is mostly about methods development. There's sort of two arms of my lab's research, the development of new methods and actually using them to solve the problems that we're interested in. Today, I'm going to be talking a little bit more about the former, but I'm going to start by motivating it by the problem that I've probably spent most of my effort on over the years. As Rui said, that is the problem detecting adaptation. And there are some who believe that this isn't really a worthwhile endeavor because adaptation is not an especially important force in evolution. One reason why I'm inclined to disagree is that life is harsh, nature is harsh, and if you're an insect out there like this little Katie did here on a leaf, if you're not careful, you're going to end up being lunch for a spider or suffer some other horrible fate or not find enough food. There are all kinds of pressures out there that organisms face, and these are constantly changing. So I'm inclined to think that adaptation is an important force because life still exists, so they must have adapted somehow. So rather than being eaten by the spider, you might want to do something like this, which katydids have done quite famously. They're very effective leaf mimics who can acquire not only the coloration and texture of leaves, some of them have holes or look like autumn leaves that have faded, missing chunks. It's truly remarkable how these things are able to blend into their environment and avoid getting eaten by predators. So it's important to adapt to the ever-changing natural world, but it's worse than that because you have to deal with humans as well. And we are very good at imposing new, very strong selective pressures on organisms, even unintentionally. We're driving many species towards extinction right now. We're in the midst of a mass extinction that's accelerating. And those are just the organisms that we are killing incidentally. We're also trying to intentionally drive some towards extinction, or at least keep the populations controlled. And this is the case for mosquitoes, like the Aedes mosquitoes showed at the top here, Egyptian albopictus, were vectors of nasty things like dengue, chikungunya, Zika, and yellow fever, and Anopheles gambiae, Culex pipiens, vectors of things like malaria, West Nile. We have strong incentive to try to keep these things controlled, so we've used a number of measures to try to do this, including insecticides. And I'll come back to that a little bit later, but just to illustrate how these organisms have coped with these selective pressures, here is some data from a group in China that was looking at the effectiveness of deltamethrin, a pyrethroid insecticide in Guangzhou, China. And over a period of a couple of years, they showed, from mosquitoes that they sampled from several different locations, that this insecticide started out being roughly 100% effective. Here's the mortality rate in 2015. And then we follow this forward just a couple of years. And for all of these locations, it's less than 50% effective. And in one, it's essentially useless. So in just a couple of years, these mosquitoes are able to get around this new, very strong selective pressure that we've imposed on them. So adaptation seems to be a ubiquitous force. And if we want to know how it happens, we need to understand what's going on in the genome, because that is the driving force of adaptation. You start at some low fitness phenotype, and then there's a mutation that every now and then you get lucky, you get a mutation that confers a fitness advantage, and it's selected on, and you go to your higher fitness state. And really, that's a dynamic process here that I skipped over, where this mutation rapidly increases in frequency in the population. Here I'm showing the frequency of the mutation over time. And we see this sort of logistic growth type of curve here, where this mutation sweeps through the population. And the first problem I'm going to be talking about today is basically about detecting these events, because they leave their sort of characteristics, their characteristic signature in the genome. So just a broad outline of the talk. I'm going to start with a description of what I call a general framework for likelihood-free inference in PopGen. But really, this is a more accurate title would be, let's use deep learning to try to solve all our problems. And it can be fairly effective for some of them, for sure. And I'm going to focus mostly on adaptation for that, but move into some other problems later on. And then we'll come back to adaptation and talk about detecting it and from time series data, a problem that I think many in this audience might be interested in. And then I'll close by coming back to our insects and see where all of this could come together in the future. So yeah, part one, the deep learning stuff. So right, we're thinking about this problem of positive selection, where every now and then there may be a beneficial mutation that sweeps through a population. So what happens in the genome when this occurs? So here I'm showing a cartoon representation of a population. It's just a bag of chromosomes here. Each black bar is a chromosome. Each gray ball is a mutation. And the red ball here is a beneficial one. So here is our starting population. This chromosome here is lucky enough to receive a beneficial mutation. So it's going to leave more offspring because it's more fit than all the other chromosomes in this starting population. So we follow this forward in time. This is what we see after some number of generations, maybe several dozen. This red mutation has increased in frequency, but it is still far from fixation. It's not reached frequency of 100%. But we're seeing some things start to occur where there's some changes. This is a crossover event or a combination event that has introduced this red mutation onto another background. So it's now found on this chromosome here that has these two gray mutations. Basically, this chromosome and this one recombined with one another. And there also may be some new mutations that occur. I'm showing this one in blue just to illustrate that it occurred after this red mutation. And I'm thinking of this as a fitness neutral mutation, but just one that introduces more diversity onto this background. So if we follow this process to its conclusion, where our red mutation has fixed in the population, at this site, the target of selection, everyone's identical. There's no diversity whatsoever. But if we move further away because of recombination and mutation events that occurred during the sweep, diversity will recover. And there are some other sort of characteristic changes. So one is the overall haplotypic diversity is reduced. So we started with 11 sequences here in this cartoon population, and they were all distinct from each other. So we had what we call 11 distinct haplotypes. And now we're down to 8 because we have 4 here that are identical to each other, 4 chromosomes that are identical. So we have only 8 distinct haplotypes. And then by the end, we're down to 5. So we've lost a lot of haplotypic diversity. Yeah, I mentioned that we lose nucleotide diversity here at the target of selection. We also get a skew in the site frequency spectrum, which is just the distribution of allele frequencies. So in beige here, I'm showing the expected site frequency spectrum in the absence of natural selection and also demographic changes. So just sort of a constant size population with no selection. Here's what the site frequency spectrum, or SFS, looks like. But when you have one of these selective sweeps, you have these hitchhiking mutations that get to high frequency, but maybe not quite all the way because of these recombination events. So we can see that this gray mutation here did not make it all the way to fixation because of this crossover. And here's another one right here that had the same outcome. So what this means is that we will have an excess of both low and high frequency derived alleles. So our site frequency spectrum becomes a little bit more U-shaped here. Note that we have an excess of mutations that are present in just one individual in our cartoon population, and we have an excess of those that are present in almost everybody. So a U-shaped SFS. And we also have an excess of linkage disequilibrium, or the association among different mutations. So basically, whenever this gray mutation shows up, the other one shows up as well. And whenever one is absent, they're both absent. And that's a consequence of this crossover event occurring during the sweep. So there are a number of different signatures that we can put together to tell us that, okay, maybe there's been a selective sweep here. And yeah, sort of the classic one that was first described by John Maynard Smith is that there's this valley of diversity around the target of selection. So if this is the coordinate of our sweeping mutation, then as we move to the left or right, diversity will recover from basically zero back to the neutral expectation. Okay, so bearing all this in mind, let's think about how population geneticists view the world and sort of view evolutionary phenomena, and how we think about how these phenomena produce patterns of diversity that we see. So in our sort of theoretical understanding of the world, we have some evolutionary phenomenon that alters the shapes of genealogies or trees. And that in turn results in particular patterns of diversity, what I'm calling our sequence alignment, the actual observation we get. So making this a little bit less abstract, if we have a selective sweep and then we sample our population at this time point right here, then the genealogy of that population might look something like this, where we've got this one little subtree here where everyone is very closely related. These are the individuals that have our beneficial mutation and therefore share a very recent common ancestor. Basically, half of our population at this point in time shares a common ancestor back here, just 100 or so generations ago. So that's why we get this genealogy. And then if you think about what this genealogy will say, well, what patterns of diversity it will result in, we will basically get, if you throw mutations down on this tree, very few mutations occurring on these little external branches here in this subtree. And what that means is that we'll have a set of individuals that are very closely related to each other, sorry, very, very similar to each other at the sequence level. And that those are these haplotypes here in the middle, which we see are very nearly identical. So that's the way we think about sort of our data being generated. But when it comes to inference, we are like detectives at the scene of the crime. We're working in reverse. So we start with our data, our sequence alignment, and we have to somehow quantify patterns of diversity from these data and then make our inference. So going back to our example, we start with our alignment, and maybe we measure something in our data like the amount of diversity or pi, and then maybe we can use that to say whether there's been a sweep or not. Though in practice, no one would use pi to say there's a sweep. There are more specialized statistics. But just to make this more concrete, pi, one of the many different summary statistics, and there are dozens and dozens of these that have been developed by population geneticists over the decades. It just sort of tells us the expected heterozygosity. Basically, if you grab two random chromosomes, how likely are they to differ? And I'm sure most of you are familiar with this, but basically, if we have our bag of genomes here, if we grab two, what's the probability that one will be red and one will be blue in this little cartoon? That's what pi is, and it's great because it's an estimator of theta. It's intimately connected to various evolutionary processes, and its sort of behavior can be nicely described theoretically. but it's not really useful for inferring evolutionary histories on its own. So now I'm getting into sort of the way I was viewing the world, and also a few other groups were maybe about 10 years ago and more recently. Instead of the idea was we could do a better job than just sort of condensing all of our data down into a single number and then using that number to infer something about evolutionary history, to instead use a vector of statistics. And I'm not going to define all of these things, but these tell us about how much diversity is there, what are the frequencies of these mutations, how much linkage disequilibrium is there, are there sort of long-range haplotypes in our sample. All of these statistics have been devised to tell us about these different patterns of diversity, these different facets of diversity. So maybe if we use all of them, we'll have more power to infer sort of the shape of the genealogy and how it has been affected by selection. And there were a number of groups doing this, and including the lab where I was doing my postdoc way back when. And I'm talking about this old work now because it's sort of led into what I'm doing in my lab right now. Here I'm just showing the accuracy of a bunch of different methods for detecting selective sweeps. In case any of you are not familiar with ROC curves, here we're just showing the true positive rate of a method, basically how many sweeps does it correctly recover, and the false positive rate, how many false sweep calls does it produce. And what you want is a method that sort of quickly shoots up towards the top left corner. That means you're able to recover most true positives without producing a bunch of false positives. And this plot is a little bit old and out of date, but I think the general result still stands, and that is that these methods up here, which are all machine learning methods that use higher dimensional summaries of data, outperform older, sort of more traditional methods that are just based on sort of one single statistic. This blue one is a thing that I developed called SCHIC. Never mind what it stands for. I'm just telling you this because we're going to use it as a foil in just a minute to compare it to another method. Okay, so rethinking our process here. Basically, instead of capturing patterns of diversity with a single statistic, maybe we should use many, and that seems to be a viable approach. I just showed you that all those methods that adopted this approach were far more effective than previously existing ones. But an idea that we had been kicking around had been to sort of skip this step. And we explored this in collaboration with Lex Flagel. And the idea was we can go sort of straight from our alignment to making our inference using this thing called a convolutional neural network, which is a machine learning tool that is designed to detect objects and images or classify images. So it can do things like facial recognition, distinguishing between different objects in an image, those sorts of things. And just to illustrate that this is a powerful approach, the problem of classifying images was a very, very challenging one just a little over a decade ago. And there's sort of a major competition in the field of machine learning called the ImageNet challenge, where your goal was to develop a method that would distinguish between different objects in different images. So correctly label this one as a mite, this one as a leopard, this one as a scooter, and so on. And this was a very difficult task for computational methods through about 2010 and a few years beyond. On this task, most methods would produce an error about 25% of the time. This is the error rate here on the y-axis, and here we're showing the winning method of the competition each year. And this was sort of like the standard for a number of years. And then very suddenly in 2012, things changed quite dramatically, and the error rate just shot way down. what changed there was the introduction of convolutional neural networks. And these got a little bit better over the years, and in just a few years, things got to the point where they were doing better than humans are able to do on these same tasks. Yes, humans sometimes get this wrong. The machines do quite well, and they got so good at this, in fact, that they had to cancel this competition in 2017 and replace it with a more challenging task. So you could say that this problem was effectively solved by convolutional neural nets. So this is sort of like the beginning of the deep learning revolution, if you want to call it that. And I don't have time to go into the details of how these methods work, but just a broad overview. Here I'm showing an actual, very simple convolutional neural network and how it is working on one input. Here our input is a hand-drawn image of an 8. This isn't a cartoon example. This is an actual thing that I drew, and you can go to this interactive tool and do it yourself and see what happens. But basically you start with your input here, and this thing tries to correctly infer the digit that you drew. And what happens is this input, this image, goes through a series of layers that we call filters, convolutional filters. And what you can see is that they all sort of highlight different aspects of the image. In practice, what these are kind of doing are detecting edges. So you can see this filter is sort of detecting edges of this shape. This one's detecting edges in this direction, and so on. There are downsampling layers, and then you repeat the process with more convolutions, and eventually things all get stuffed into this big vector, and it gets to the point where it's pretty uninterpretable. Towards the top of the network, you can start to see, you can see what's going on, but eventually it starts to look unreadable to the human eye. There is signal in here that the neural network is able to make sense of, and we can see that it's correctly said that this is an 8 here, if you look very closely. So that's how CNNs work. But what if this is our image, our alignment of our sequence data? Or in population genetic context, where maybe we're dealing mostly with biallelic data, we can just think of this as a black and white image where white is the ancestral allele and black is derived, or vice versa. So if this is our image, how well can we solve this problem of detecting selective sweeps in the genome? And here I'm showing the results of that approach by comparing it to that Schick method, which in 2016 was right around the state of the art. And it consists of, I skipped over what Schick does, but basically it's a set of statistics developed over 80 plus years of theory by the leading experts in population genetics. And based on that vector, we try to detect sweeps. The CNN just takes our alignment, shoves it into a neural network, and trains it on the basis of simulated data. Both of these things are trained by looking at simulations. And here I'm showing the results of these two. So this is something called a confusion matrix, which is not supposed to be confusing. It's to show how confused our method is. And the idea is that we show the fraction of examples that were neutrally evolving, so there's no sweep, and the fraction of those that were correctly predicted to be neutral. The same thing for selective sweeps. I'm looking at two types of sweeps here, hard sweeps and soft sweeps. Never mind the distinction between those two. That's not essential for this talk. But the point is, diagonal elements here are those that are correctly classified, and you want these to be high. Off-diagonal elements tell us about the different types of errors that we're making. For example, Schick will call 20% of our soft sweeps incorrectly as neutral. So some false negatives there. So anyway, diagonal elements, we want those to be high. And what you can see here is that the diagonal entries are all higher for the CNN than Schick. It's got greater sensitivity to detect selective sweeps, and it has a lower false positive rate. So it's better in every way, even though it knows nothing about population genetic theory. It's just looked at some simulations and done its best to sort of learn what patterns to detect. So this is a result that we found really exciting. And we were thinking, about how we can maybe apply this to other problems in population genetics. And I'm just going to go through some of these very quickly. First, I want to say that it can be used not only for classification tasks, what we would call model selection in evolutionary genetics, but also for inferring real valued parameters like recombination rates and demographic parameters. And it does this quite well, but I'm going to gloss over that in the interest of time. So another question that we're going to spend some time on in this talk is detecting introgression. And this is relevant because speciation is often followed by gene flow. If we have some ancestral species here that splits into two, there is often secondary contact where these two species will hybridize. And often this comes with a fitness cost because you will get combinations of alleles that have never showed up in the same genome before, and they may be incompatible, and this will result in lower fitness. And every now and then introgression is adaptive. So a really fun example is from heliconius butterflies, where there are different species in different parts of the tree that have nearly identical wing patterns. And this isn't convergent evolution. This is because these species have hybridized with each other and through gene flow acquired these patterns. And these are adaptive because they basically signal to predators in the area that you shouldn't eat me, I don't taste very good. Even though that can be a lie. If one species has evolved to be sort of toxic and the other one enters the area, then it can sort of steal, it can cheat by stealing that wing pattern and fool predators. So it's a sort of quick route to adaptation if you can steal from your sister species that has already adapted. Okay, so this is something that I also worked on during my postdoc a little bit. Again, taking a classification approach where we're sort of moving across the genome and trying to distinguish between three different models. A model of no introgression, a model of gene flow from one species, from species 1 to species 2, or from species 2 to species 1. And here our two species were simulins and Seychellia and Drosophila. And we created this method called fillet. It stands for something, but I'm just telling you the name so I can compare it to something else in a moment. And we showed that fillet works very well compared to existing methods. Here's sort of the range of parameter space, basically the intensity of gene flow and the timing of gene flow, where fillet has power to detect introgression compared to univariate statistic methods. Basically, we've dramatically expanded the parameter space where gene flow is detectable, and that's great. But the CNN does a lot better. So again, if you look at the entries along the diagonal, we're able to produce far fewer false positives and a lot more true positives here. So again, using a vector of statistics that have been designed to find the patterns of gene flow doesn't work quite as well as feeding a bunch of examples to a neural network and asking it to sort of learn its own statistics, which is effectively what it's doing. So something that we're working on right now, this is unpublished work, but sort of wrapping it up, and that is going a step further and trying to not just figure out if there's been some gene flow in a part of the genome, but which individuals are migrants or descendant from migrants, I should say, and which parts of their genomes are introgressed. So basically pulling out the introgressed haplotypes. So here's our input representation. Here we have a two population alignment, and we are here visualizing which haplotypes are introgressed. So sort of purple means not introgressed. So these are all individuals in population 1 that trace their ancestry to population 1. These are individuals in population 2 that have ancestry from population 2, but these yellow ones here are individuals in population 2 that have ancestry from population 1. So they're products of gene flow. And our goal is to produce this type of prediction where we're able to, as closely as possible, match the true history of introgression. And this is an actual prediction from our method where it's just about nailed this thing. So how does it work? So this is work by a computer programmer in the lab, Dylan Ray, and what he did is develop this thing called a UNet, which has been used for this problem of semantic segmentation, where you're not just trying to classify an image, but you're trying to say which pixels in the image belong to a person, which pixels belong to a bicycle, and which are neither. So making a prediction for every single pixel in your image. And the way this works is you sort of start with the standard convolutional neural network approach of repeated convolutions and pooling. But then you have the second half of the network where you do deconvolution and upsampling, where basically you go from a lower dimensional space back up to an output that is the exact same shape as your input. So you're making a prediction for every single input pixel. And there are some other sort of key points in the network that allow this to work, but I can't really go into all of them in detail. But the point is that you are able to make a prediction for every pixel. In our problem, that means we're able to predict each individual at each SNP as either introgressed or not introgressed. And it seems to work remarkably well. So stay tuned for that. We're writing that up. Okay, so now I'm just going to jump into an entirely different field, but it's related because we're using, this is related work because we're using a similar method. So this is what Anton Suvorov, a postdoc in the lab, has been working on. And his first project in the lab was to basically adapt this same approach to inferring phylogenetic trees. So if we have three species here, there are three possible tree topologies, one where A and B are sister species, one where A and C are sisters, and one where C and D are sisters. I'm talking about different species, but really this is the problem of inferring gene trees, which is not necessarily the same as the species tree, and we'll get to that. But can we do this just by shoving this input alignment into a neural network? And the answer is yes, it works quite well. So how well it works depends on the parameter range that you're examining, but across a sort of broad range of parameters, this method performs about as well, if not better, than sort of standard state-of-the-art methods like maximum likelihood and Bayesian inference. So that's encouraging, but what's really exciting about this approach is that it's really fast, at least once you've trained it, and it trains pretty quickly too. But if say we're trying to infer an unrooted quartet topology, so that three-class classification problem, if we wanted to do this with exabays using a 10 kilobase alignment, it's going to take about 3 minutes to do this. If we're going to do this using our convolutional neural network, our prediction will be completed in about a millisecond. So, and that's because you can parallelize these things on GPUs. The neural networks are very fast when they make their predictions, basically. So this is an exciting prospect because there are many steps of phylogenetic inference that are quite slow. And if we can use deep learning to speed some of that stuff up, that could be a real practical benefit. And there are some other groups working on this as well as ours. So we're sort of excited to see this work taking off. So something that Anton is working on right now is extending this approach to infer branch lengths as well as the tree topologies themselves. And it's working quite well. We're able to recover true branch lengths generally with higher accuracy than maximum likelihood, which is sort of the standard method for doing this. And we're thinking of maybe taking this approach and applying it to some data that we've put together of about 150 different species from the genus Drosophila, and we've inferred a phylogenetic tree for this genus for the genomes that we sampled from this genus. There's still over 1,000 more yet to be assembled. But we were able to produce a very deep and we think fairly accurate phylogeny. This was not using our deep learning stuff because it is not able to handle such large number of sequences yet. But we might be able to refine these branch lengths using this approach. But really the reason I'm mentioning this is because it gives me a chance to talk about this other project, which I thought was a really fun one with an interesting result, especially for Drosophilists. And that's related to introgression. So we've been talking about inferring gene trees, the history of each sequence along the genome, but this may not necessarily match the species tree. For example, if there's been introgression, that will result in different topologies. So if our species tree is A, B, and then C, if B actually traces its ancestry for some genes back into C because there was introgression going forwards in time from C into B, then for those genes, we're going to have a different tree. C and B are going to be sisters. And we can get the same thing if there's introgression between A and C, where A and C are sisters. So what this means is that for some parts of the genome, you will get different tree topologies and also different branch lengths that can give us a clue that there's been some introgression. And this was something that we examined across this phylogeny. And we're kind of limited in how well, how sort of clearly we can paint the picture of introgression across this phylogeny. Here we split it up into 9 different clades and are looking within each clade. And really what we can do is test to see which pairs of species are introgressed. So these triangular matrices are showing different pairs of species and showing which ones we think have strong evidence for introgression. And then on the basis of these patterns, we show these arrows, which we think are sort of the best interpretation of these results. So this is sort of like our best guess at where gene flow has occurred along this tree. Let's take a closer look at one of these. So here we see that there's evidence of gene flow between Obscura and Bifasciata and this group of species, Aphanis, Athabasca, Azteca, and so on. And there are a number of introgression histories that are consistent with this observation. We've just sort of used a parsimony type heuristic to draw these arrows and say this is all probably one gene flow event that occurred between the ancestors of these 2 clades. And that's how we get this block here. So that's what we get. Sometimes this heuristic will draw some things that look a little bit weird. And I'd be happy to chat about that later if there's time and you'd like to hear some questions about it. But I think it demonstrates that there's some limitations here. And we're trying to get around this by thinking about how to better disentangle all the different possible histories of introgression. But there's a very massive number of these. Basically, this is the number you get if you consider introgression between all sampled lineages. This is not even accounting for ghost introgression, where you could have introgression between unsampled lineages in your tree, which could also affect your data. So yeah, this is an astronomical number of different histories if we think of the presence or absence of each of these independent possible gene flow events. But we're playing around with using a deep learning approach to sort of simultaneously make a prediction for each of these possible gene flow events. And this works very early on, so I can't tell you if it works or not, but a student in the lab, Josh Sichter, is working on it, and I hope to, in the near future, be able to say something about how well it has worked. Okay, so reached the end of part 1. Don't worry, that's the longest part by far. The conclusions up to this point are simply that, yes, deep learning can work for helping to solve a number of problems in population genetics and phylogenetics as well. I think one of the main strengths of this approach is its flexibility. Basically, if you can produce training data, often through simulation, then you can try to solve the problem. You don't need to have had all of the theoretical development of statistics tailored to this problem. The network can learn its own. So it's very versatile, and that's great. And just one quick aside is that you can do more than just use these networks to take data and make inferences, go from data to a model, you can also do the opposite. So here's some work that's being done by Will Booker, a postdoc in the lab, and it's inspired by this person here. Does anyone know who this is by any chance? Speak up if you do. Okay, I'm not hearing anything, and that's good because this is not a real person. And the idea here is that neural networks can generate data. And I don't have time to go into the details of how these work, but basically you can create a generator network that produces images, or in our case, alignments, and try to train it to fool a discriminator that discriminates between real and fake images. And over time, these get better. And it turns out this works quite well for producing artificial genomes, which is pretty cool. And there's some other labs working on this as well. But I have to sort of skip all of that. Okay, so part 2 is delving into time series. And of course, everything I've talked to up to this point assumes that we have data from a single time point, the present or the very, very recent past. And of course, wouldn't it be better if we had data from many different time points? This is a feeling that I think many in this audience are sympathetic to. And our idea here was to develop a method that would be useful for using time series from natural populations. So we don't have replicates available, but we have repeated samplings from one single population. And it was sort of inspired by trying to capture all the information you might see in a molar plot, but in a recombining organism. And we experimented with a few different approaches, but what we settled on was one where it was just sort of looking at the trajectories of allele frequencies within a window of the genome. So just sort of zoom in on one chunk of the chromosome. Here is on the y-axis, we're showing our position. So these are different SNPs, different polymorphisms. And the starred SNP here is our focal one. In this case, it's an allele that's under positive selection. And we're just showing changes in frequency. And we can see that this thing has increased in frequency over time. So this image here is actually the average of many simulations. So on average, the sweeping allele increases in frequency. On average, these other alleles don't do anything. Sometimes they go up, sometimes they go down. So we're sort of missing a lot of the interesting dynamics here. But our goal, you don't see that in the average, but we're training this thing on many, many different simulated examples under different selection coefficients or combination rates. And the idea is to expose the method to a large number of these hitchhiking events such that it will learn sort of the joint distribution of allele frequency trajectories in a region surrounding a sweeping mutation, and to be able to distinguish that from neutrally evolving ones. So if you look at all these examples, you see that none of them look exactly the same. None of them look like your sort of textbook cartoon version of a selective sweep. They're much noisier, but you can see that, okay, there are some sort of hitchhiking alleles here, and I don't really see that in the neutral case. And deep learning is a great approach for trying to capture these patterns. So this is an approach that we tried out. Basically, we simulated a bunch of data using SLIM. We generated those input images that I just showed and plugged them into a convolutional neural network. And what we found is that it worked quite well. It worked better than some other approaches we tried, and also previous approaches from some other groups. So this green line is showing an approach where you're just sort of looking at allele frequency increments, so changes from sampled time point to time point over a whole series. So that examines just a single SNP, and we're sort of doing the same thing for a whole region and sort of considering the joint history of all of those polymorphisms. So we're able to do a bit better there, which is encouraging. What I think is really exciting about this approach is that it's able to localize the target of selection quite accurately. So basically here, we're showing over a set of simulated examples where there's a sweep in the center of our simulated chromosome. Where do we say there was a sweep? And we almost always say there's a sweep at the center. but there's no sort of shoulder effect that often affects sweep detection methods where you have trouble localizing it. We're sort of able to produce a very narrow peak on average, which is very nice. So this thing seems to work quite well. We also did test it out on data from the Schrider lab, and we found that it produces highly replicated peaks across 10 different replicates. So that's another great sort of sanity check using both simulated data and experimental data. We see that this thing works because it uses just allele frequencies. It is applicable to pool sequencing data, as well as data with phased haplotypes or anything in between. And it's a very flexible software tool that that Logan has engineered. So that's something that I'm very happy with, very proud of his work on that. Okay, so just to sum this up, time series data are very useful, preaching to the choir here, but I think deep learning is potentially a very useful way for trying to interrogate these data. Okay, so last but not least, I just want to quickly touch on some work we're doing in Aedes. This is work we're doing in collaboration with Daniel Matute, and this is being led by Becca Love, a postdoc in the lab. And these Aedes mosquitoes, they're a real troublemaker, not just because of everything that they vector, but also because their ranges are expanding quite dramatically. Both Aedes aegypti and albopictus, their ranges are predicted to expand quite a bit over the next few decades. So they're going to become more of a problem for more people. Yeah, here I'm showing their range expansions in the United States because I'm American and we only think about ourselves, of course. So, but this is a global phenomenon. So we examined data from a number of different locations, including 2 locations in Colombia that we sampled and sequenced. And we were interested in looking at things like population structure, but I'm just sort of going to gloss over that to get to the fun stuff, which is natural selection. And recall that we've imposed some very strong selective pressures on these bugs. What's going on in these? So we know very little about the demographic history of these species. So I wasn't feeling super comfortable in using a highly parameterized model of selection. So we went old school with this and just used this approach called Sweep Finder, which looks at changes in the site frequency spectrum surrounding a selective sweep. If you have a sweep, basically you expect to have your U-shaped SFS, as I described earlier, and as you move further away, you recover towards sort of the normal expectation. Sweep Finder is now sort of a classic method for doing this, and I think it works quite well, especially for very strong selection, which is what we're looking for here. So no need for the fancy stuff yet. So here we're showing our results, and on the y-axis I'm showing the Sweep Finder's composite likelihood ratio, and we're showing values across each chromosome, the 3 chromosomes of the Aedes genome. And I want to put these values in some context here. Note the scale here on the y-axis. So I'll come back to those in a second. But first I want to show a top candidate for selection from humans. This is one of the strongest selective sweeps in humans. It has a composite likelihood ratio of about 22. And here's the scale here in Aedes. And these aren't exactly apples to apples comparisons. Every time you run this on a different data set, the composite likelihood ratios will be different. But I still think the fact that we're getting ratios in the thousands is pretty dramatic and should suggest that there's super strong selection going on here. I would say that this little tiny peak here is probably very strong selection, but it looks very small in comparison to these signatures here. So these genes that I'm highlighting have been shown to be under strong selection and to show evidence of conferring insecticide resistance in other insects. And Vssc, the voltage sensitive sodium channel, has also lots of experimental evidence in Aedes as having evolved insecticide resistance. And we know it's a target of things like DDT and pyrethroids. So we're going to take a closer look at that one, the really big sweep. So looking at South American data, previously sequenced Brazilian data, and our Colombian data, we see a difference here. We see very strong signatures of selection in both, but the sweep signature is larger and broader in Brazil. So why is that? One thing that we noticed is that if we look at our data in principal component space, so here's PCA of our sort of global sample here from 6 different countries. If we focus just on the Colombian samples, we basically have these 3 different clusters, and these data are just from the Vssc region. So we just plug that gene into PCA to see how things look there. So our Colombian data look very different in terms of how clustered they are relative to the other sampling locations. And we also noticed that there were a number of locations where the contribution of these different SNPs to the location of examples in principal component space was especially large, especially along the second principal component. And that's the one that divides our Colombian samples here. And these locations here correspond quite nicely to several known insecticide resistance mutations, some that have been shown experimentally to have effects, others that are associated with resistance and hypothesized to have effects. And I'm going to skip over this, the details of staring at this, but basically we find in our data, there's some missing data, but if you look closely enough, you can see that it appears that these 4 additional insecticide resistance mutations are in perfect LD with each other in our Colombian sample. And there's this fifth mutation, F1534C, that is fixed in Colombia, as well as in Brazil. So if you look at those 4 additional resistance alleles, this top cluster corresponds to individuals that are homozygous for all 4. The middle one is heterozygotes, and the bottom is homozygous for the wild type at all 4. So your genotype here determines where you cluster in this region, and it appears that these are 2 distinct haplotypes, and these are the heterozygotes. Okay, so what's going on here with respect to our difference in Colombia and Brazil? We see that there's very little diversity if we look at both the top cluster and the bottom one in Colombia, and yet our sweep signature is weaker than in Brazil. So here's what we think the explanation is. Basically, in Brazil, we had this selective sweep where this F1534C allele swept through the population, and that same sweep probably occurred in Colombia. But then some other haplotype emerged with these 4 additional resistance mutations. Where did it come from? Don't know. But it showed up in Colombia, and then it started sweeping, and it's not a completed sweep yet. So we have this haplotype at intermediate frequency. So we have an excess of intermediate frequency alleles there, and that's sort of shrouding our signature of selection, which is why it's been partially eroded relative to Brazil. So what we think is that there's been massively strong selection at multiple loci in these populations, especially at the Vssc gene, but an potentially ongoing selective sweep of 4 additional insecticide resistance mutations that are all in perfect LD with each other has partially eroded the signature in Colombia. So we have a lot of questions about this. Of course, what about loci under weaker selection? I highlighted that one little weaker peak on the left. Can we say something about what's going on there? And we're very interested in doing that. But what about this haplotype of 4 resistance mutations? Is this like an uber resistance haplotype? Where did it come from, and what's going to happen next with it? So this is something that we're going to try to address in the future, and this is sort of where all the different parts of the talk come together. Our plan is to do repeated sampling of these locations from Colombia and see how these mutations have been changing in frequency, and do the same approach genome-wide, and basically adapt that time sweeper method, which I described in the previous part, to these data from Aedes. And that might give us more power, not just to detect weaker selection across the genome, but also figure out, what is going on with this haplotype. Is it going to continue to sweep, or has it potentially reached some equilibrium frequency, perhaps because of trade-offs of, all of these amino acid replacements in this very important gene? There's a trade-off between providing resistance and, not breaking your voltage sensitive sodium channel so that you can still have proper neural function. So it's a very essential gene. So by continuing to monitor these changes over time, maybe we'll be able to figure that out. And we're also going to try to sequence some samples collected in the past to sort of put that all together. So this is work we're really excited about, but it's in the very early stages. So stay tuned for that. And that's really sort of my conclusion side, is, that all of these different things I talked about today are hopefully coming together into something. So with that, I'll acknowledge the people that did the work, including those in my lab, past and present, and collaborators and funding sources, and of course you all for having me here and for your attention. And I'd be happy to take some questions.